girls, welcome to video number 4. This is on the Kreb cycle. Now again, I'm going to go into quite a bit of detail during this video, but at the end I'm going to bring you in and tell you exactly what you need to be able to remember. So I'll give you the nice big picture, but then there's a list of things that make it a little bit easier that we have to remember. So we're starting off from where we finished at the end of glycolysis, but now we're in the mitochondria. So this is occurring in the matrix of the mitochondria. And I'll show you why in a second. But that's really important that we know that we are in the matrix of the mitochondria right now. We're starting off with the pyruvate from glycolysis, and we've got 2 of those. And then each one of them goes through this big biochemical process, biochemical pathway. This is the detail you don't need to know. And that is called the Kreb cycle. Sometimes we also call it the citric acid cycle. We're going to come back and talk about this other coenzyme we talked about before, so that's NADH. Again, no P because we're not in photosynthesis, so just NADH. And we're going to be making a lot of that. We're also going to be talking about this guy down here, carbon dioxide. So here is the next one in the series of videos from the fabulous Glenn Wokenfield. He does amazing work, but again, way more detail than what you need to know. But I want you to click on the link and make sure that you watch him because it is good fun. Okay, oh, sorry, it's wanting to play. So this bit here is too much detail, but I will be walking you through it just so that you can hear the story, and then we'll focus on the bits you need to remember. So we are taking place in the mitochondrial matrix. The pyruvate that's come from glycolysis is going to diffuse into the mitochondria through that outer membrane, and then it diffuses through the outer membrane. Then we actively transport it into the inner membrane, and then we convert it to this material called acetyl-CoA and 1 carbon dioxide. Okay, way too much detail, but I'm going to show you what's happening here. So here's an animation that I made, and I often find that animations can be a really good way to prove that I understand what's happening. So you can see up the top here, we start up here, they've got 2 pyruvate molecules. Now what they like to do is they like to diffuse from the cytoplasm through the outer membrane, and then they get actively transported. So what are we going to be using? We're going to be using ATP here, into the mitochondrial matrix. A whole bunch of reactions happen and we end up with some carbon dioxide and some acetyl-CoA. But what can you notice over here? We've still got, up in the top left-hand corner, we've still got some pyruvate. So it's going to happen again. The pyruvate's going to diffuse through the outer membrane, get actively transported through the inner membrane, get converted into acetyl-CoA and a carbon dioxide. Okay, after that, it needs to go through another turn of the Krebs cycle, and we're going to produce another 2 carbon dioxide per pyruvate. So let's see how we go. So here is the Krebs cycle in here. You can see that we've got 1 carbon dioxide over here, 1 carbon dioxide over here, but we need to end up with 6. So what we do is these 2 acetyl-CoA, we'll start spinning through that Krebs cycle, and each one will create 2 more carbon dioxide. So here's the other one, coming through, make our other 2 carbon dioxide. So what have I got left? 1, 2, 3, 4, 5, 6 carbon dioxide. Okay, in addition to this, the Krebs cycle makes 6 NADH. Now you don't need to remember that number. 2 FADH2, which are not even on your study design, but you will see them when you are reading, and 2 ATP. Whoa, look at all the amazing coenzymes that we've loaded up here. Here is a nice confusing diagram just to show you exactly what's happening during the Krebs cycle. I haven't even studied this. I have no idea what the names of any of these are, so please do not waste any of your time thinking about it. But this is another example of a biochemical pathway. You can see that the product of one reaction becomes the reactant or the substrate in the following reaction. The product of this is going to become the substrate for the next reaction, and it follows along. And the reason it's a cycle is because this last molecule ends up becoming the one that it was at the beginning, and it just kind of like spins around and around, but you don't need to know any of those names. This is what you need to remember. You must remember it happens in the mitochondrial matrix. Okay, so I'd highlight this part in my notes. You must remember what goes in and what comes out of each section. So what we put in was the pyruvate. Where did the pyruvate come from? The pyruvate has come from glycolysis. Okay, what do we get out as a result of that? We get carbon dioxide out. Can you see how I've put the numbers of each one in brackets afterwards? So you can see I put in 2 pyruvate and I get out 6 carbon dioxide. Now you don't need to remember those numbers, but some people, if you're mathematically minded, if you want to do the chemistry side of it, they might help. What they are looking for you to remember is that pyruvate goes in and carbon dioxide comes out. In addition to that, while we're breaking down this pyruvate into carbon dioxide, this carbon dioxide is a waste product. So we don't really want that. So why did we bother going through this whole Krebs cycle? The whole point of this Krebs cycle is to get these guys and these guys. So the NADH, we also made NADH in glycolysis. The NADH for this is going to be needed in our next step. The ATP can be used for energy for any one of those processes that we talked about in our introductory video. So you can see that the chemical that we've made here isn't what we really want from this. What we really want from this is the ATP as stored form of energy, and also that NADH for the next stage along.